This is human brain tissue inside a rat brain. Some scientists think that it could help with understanding the human brain, and one day even treat conditions like schizophrenia, depression, and autism. But this tech opens up a lot of ethical questions. That green blob is called an organoid. An organoid is a ball of cells with certain characteristics of an organ. So in this case, the brain. In lab dishes, brain organoids look like little pearls. Researchers study them because they mimic how human brains develop and how they work. They're grown from a person's skin cells that then can be engineered to turn into any kind of cell in the body. And that opens up the door to potentially being able to develop specific treatments for specific people. But organoids grown in a lab dish have their limitations. Unlike a real living brain, organoids don't have blood vessels to feed all their cells, and that limits how big and complex they can get. They also don't get information like touch, vision, and smell that a real brain does through our hands, eyes, and noses. Scientists implant organoids into animals because it allows them to study brain disease in an environment that's closer to a normal brain. These new hybrid brains are called chimeras. This recent Stanford study found that neurons in chimeras grew more than neurons in organoids. See this small, stumpy neuron? That's from an organoid growing in a dish. This one on the left, it's from a chimera, and it's way bigger and has far more branches. And this is how neurons normally look in the brain. Those branch-like tentacles make connections to other neurons and form huge networks that are at the heart of how the brain works. Now, this is important because brain conditions change these networks. The Stanford team said using chimeras helped them discover key differences in cells that make up brain networks. They take organoids made from cells of patients with a genetic condition called Timothy syndrome, which is associated with autism and epilepsy. Scientists then transplant those organoids into the brains of baby rats. They found the neurons in these chimeras had less branching and were smaller than in chimeras with healthy organoids. Remember that neurons in dish-bound organoids are also less complex. Without chimeras, the researchers may have missed this difference. This highlights the advantage of using chimeras for understanding brain diseases and coming up with new treatments. That's why some scientists are so excited about brain chimeras. But one big question is whether human neurons actually hook up to rodent neurons. If they do, you'd expect their activity to affect the animal's behavior.
The behavior output is the ultimate proof that the organoids are really part of the entire system.
The same Stanford researchers grew organoids with healthy human neurons that had been tweaked to have sensors that respond to blue light. They flashed pulses of blue light directly into their chimeric rat brains through a fiber optic cable. This is important. The rats couldn't actually see the blue light with their eyes. The flashes were literally all in their heads. By pairing the light pulses with a sip of water, the rats learned to expect a drink whenever the light flashed. That lesson was wired into the human neurons with the blue light sensors. So eventually, when the researchers flashed blue light into those neurons, the rats went looking for water on their own. And what that shows is that the organoid had become a working part of the animal's brain. The Stanford study only looked at behavior in animals transplanted with healthy human organoids. The next step will be testing behavior in animals implanted with organoids from patients with brain conditions and seeing whether that's different. That kind of information would get scientists closer to coming up with better treatments. Chimeras are an incredibly useful tool, but they have some limitations. First, they are very labor-intensive, which makes scaling them up difficult. And animal bodies are not the natural home for human brain tissue, which could affect how neurons grow. And then there's the big ethical questions, like do chimeras have human-like qualities? Human organoids integrate pretty well into rodent brains, especially if the transplant happens when they're babies. That's when brains are most susceptible to change, so humanized chimeras are a possibility. So far though, scientists say they haven't seen signs of higher intelligence or other human qualities. And that leads to another important ethical question.
Could we be creating unintentional suffering without knowing that we're doing so?
The Stanford researchers didn't see signs of memory loss, seizures, or increased anxiety. But as in any experiment, it's always possible to miss something important. So basically, scientists have to figure out whether they have to think about chimeric research more like human research. Brain chimeras are only one example of a growing number of high-tech and somewhat controversial strategies to study and combat human diseases. That includes human monkey embryos and pig organs transplanted into humans. Researchers have even restored some function to dead pig brains. With all of these, society will need to balance the promise of better therapies with the very real ethical concerns these technologies bring up.